Hello, welcome to Biology, the scientific study of life. Many proteins work together in DNA replication and repair. The relationship between structure and function is manifest in the double helix. The idea that there is specific pairing of nitrogenous base in DNA, bases in DNA, was the flash of inspiration that led Weston and Crick to the correct double helix. The specific principle base pairing to a template strand. In a second paper, Weston and Crick stated their hypothesis for how DNA replicates. Now, our model for deoxyribonucleic acid is, in effect, a pair of templates, each of which is complementary to the other. We imagine that prior to duplication, the hydrogen bonds are broken and the two chains unwind thus and separate. Each chain then acts as the template for the formation into itself of a new companion chain, so that eventually we shall have two pairs of chains where we only had one before. Moreover, the sequence of the pairs of bases will have been duplicated exactly. Notice also a model for DNA replication, the basic concept. In this simplification, a short segment of DNA has been untwisted into a structure that resembles a ladder. The rails of the ladder are the sugar phosphate backbones of the two DNA strands. The ranks are the pairs of nitrogenous bases, simple shapes, simplified. The four kinds of bases, dark blue represents DNA, free nucleotides, and neatly synthesized DNA. Notice that the parent molecule has two complementary strands of DNA. Each base is paired by hydrogen bonding with its specific partner, A with T, G with C, and so on. Notice that. The first step is in replication, is separation of the two DNA strands, and notice that each parental strand now serves as a template that determines the order of nucleotides along a new complementary strand. Notice also third one or fourth one, the nucleotides are connected to form the sugar phosphate backbones of the new strands. Each daughter DNA molecule consists of one parental strand and one new strand. Notice also this figure illustrates Weston and Crick's basic idea. To make it easier to follow, we show only a short section of a double helix in untwisted form. Notice that if we cover one of the two DNA strands in this figure, we can still determine its linear sequence of bases by referring to the uncovered strand and applying the base pairing rules. The two strands are complementary. Each stores the information necessary to reconstruct the other. When a cell copies a DNA molecule, each strand serves as a template for ordering nucleotides into a new complementary strands. As nucleotides line up along the template strand according to the base pairing rules, they are linked to form the new strands where there was one double-stranded DNA molecule at the beginning of the process. There are soon to each an exact replica of the parent molecule. The copying mechanism is analogous to using photography. analogous to using photographic negative to make a positive image, which can in turn be used to make another negative, and so on. This model of DNA replication remained untested for several years following publication of the DNA structure. The request experiments were simple in concept but difficult to perform. Weston and Crick's model predicts that when a double helix replicates, each of the two daughter molecules will have one old strand derived from the parent molecule and one newly made strand. This semi-conservative model can be distinguished from a conservative model of replication in which the parent molecule somehow reforms after the process, that is, it is conserved. In yet a third model called the dispersive model, all four strands of DNA following replication have a mixture of old and new DNA, and the next page of mechanisms of conservative or dispersive DNA replication are not easy to devise. These models remained possibilities until they could be ruled out. Finally, in the late 1950 years, Matthew Michelson and Franklin Stoll devised experiments that tested the three hypotheses. Their experiments supported the semi-conservative model of DNA replication as predicted by Weston and Crick. The basic principle of DNA replication is elegantly simple. However, the actual process involves some complicated biochemical gymnastics. Notice that three alternative models of DNA replication. The short segments of double helix here symbolize the DNA within a cell beginning with a parent cell. We follow the DNA for two generations of cells, two rounds of DNA replication. Newly made DNA is light blue. Notice that. A parent cell and first replication and second replication, this one. Conservative model, the two parental strands reassociate after acting templates for new strands, thus restoring the parental double helix. Notice that. B semi-conservative model, the two strands of the parental molecule separate and each functions as a template for synthesis of new complementary strands. Notice that. Notice also dispersive model. Each strand of both daughter molecules contains a mixture of old and new and old and newly synthesized DNA. Okay, thanks for listening.